Good afternoon, everyone, and welcome to today's Alum Zoom. We are today featuring Dr. Jennifer Israel. She is Senior Genomics Data Scientist at Kriya Therapeutics. My name is Melissa Bostrom, and I'm the Assistant Dean for Graduate Student Professional Development in the Graduate School. And I want to welcome you to today's session on behalf of the Graduate School and our co-sponsor, the Office of Postdoctoral Services. Let me begin by introducing our speaker. Jennifer Israel, PhD, PMP, is Genomics Data Scientist at Kriya Therapeutics. That just rolls right off the tongue. Yeah. She received her PhD in Genetics and Genomics from Duke University in 2015 and subsequently completed a training experience as a postdoctoral scientist in computational genomics at UNC Chapel Hill that ended at the beginning of 2018. She then transitioned to her first position in industry as a scientist data team lead for Novozymes, where she spent about 2.5 years. And she followed that role with a position as a computational biologist for gene editing with Sarepta Therapeutics. And then in April 2021 began her current role with Kriya Therapeutics. We are delighted to welcome her back to campus virtually to learn about her career path in more detail and to hear about the open positions for PhD level scientists at Kriya. Please join me in a round of virtual applause for our featured guest today. So for any of you who have not joined us for previous sessions, we'll begin with the 5 career questions that we ask all of our guests, with a bonus 6th question today about those hiring opportunities at Kriya Therapeutics. And then we'll go to questions that come in through the chat, or if you'd like to raise your hand and ask a question, and we'll move to those questions for the end of the session. So with that out of the way, I'll turn to Dr. Israel and ask, could you tell us a bit about your background?
Sure. So I have an undergraduate degree in biology, and I came to Duke through the developmental and stem cell biology program, and I affiliated with UPGG, which is the Genetics and Genomics program at Duke. I was co-advised by Greg Ray and Dave McClay, and my thesis research was on evolutionary genomics in non-model echinoderm species, or not like sea urchins and starfish. And I was really into development and evolution. And I started out thinking that my PhD was going to be mainly a wet bench thesis project, microscopy and working with embryos, in the lab. But I started a side project that was building de novo transcriptome assemblies. And so I got really into the genomics side of research and turned my PhD around, ended up being fully computational. And then I felt like I needed more training. And so I went to UNC with Terry Fury, and that's a fully dry lab. So working with a lot of people who had backgrounds in computer science, and did more comparative genomics, but looking at questions at least that were a little more translational. So not really evolution, but I was doing like pollutant exposures and different mouse lines, and a little bit of work with human data. And I knew when I started my postdoc that I wanted to go into industry, and Terry knew that too. And so we kind of said, okay, a good postdoc time is around 2 to 3 years, if you know that you're going to end up going into industry. So I did 2 years, and then I went on the job market. And it's kind of funny the way things worked out, because I applied to a lot of positions that were bioinformatics based, but the offer I ended up accepting was not a bioinformatics role. It was not a genomics role. It was a data science role, working in product development. So not even in R&D. It was very translational. At the end of the product development pipeline, all the R&D had been done. They were looking at kind of product performance and product stability. And I learned a lot in that role. But after 2 years, I kind of realized that I wanted to get back into R&D. I wanted to get back into genomics. And so I went to Sarepta for a while, and then I took a new opportunity at Kriya. And so that's where I'm at today.
Wonderful. And thank you for that level of detail that you shared. So fast forwarding now to your current position, can you tell us a bit about how you got that job?
Yeah, so I was going to tell you guys a couple of different ways I've gotten jobs, because each position I've gotten, it's happened a different way. So my first job out of my postdoc at Novozymes, that was just me applying, I think through LinkedIn or through Indeed, and kind of the normal route. And then for my Sarepta position, when I was at Novozymes, I kind of decided, okay, I want to join a company that's in the human health space. I want to do genomics. I don't want a large company. Novozymes, I think, was 7,000 employees. I wanted a small company. I wanted it to be US based, have a presence here in RTP. So that kind of like narrowed down a lot of the search space. And I had seen that Sarepta was hiring for molecular biologists and like bench scientists. And based on those job descriptions, I knew that they would be generating data at some point that would make sense for them to hire in someone with my skill set. And so I proactively reached out to them and said, this is my background. This is what I want to do. It sounds like you may, have a need for this down the line. And it turned out they did have a need, and they just hadn't advertised it yet. And so that's how I got my job at Sarepta. And then for Kriya, Sarepta had undergone a reorg early last year. And I wasn't particularly thrilled with how the reorg went. And I wasn't seriously job searching, but a friend of mine from Duke, from graduate school, had just started at Kriya as a project manager. And I just reached out to her saying, how do you like Kriya? I may be looking for something 6 months from now, depending on how this reorg goes. And within a day, Kriya had emailed me and said, oh, we want to interview you now. So, and it was just an incredible opportunity that I couldn't pass up. So each job I kind of got a different way.
I love those stories of proactively networking and just leveraging those relationships to get information and build your career. Wonderful. So thinking about your role at Kriya now, can you tell us what a typical day is like? And I guess you're recent enough at Kriya that you didn't have a ton pre-COVID.
Yeah.
Know what the before times were like there.
Yeah, so I don't know what the before times were like at Kriya. The company was founded in late 2019, so they didn't even have much pre-COVID time. And, the typical day, I have a lot of project team meetings. We have a lot of different programs in the pipeline. We have, oncology targets, we have ophthalmology targets, metabolic diseases that we're going after. And so a lot of my time is spent in project team meetings, talking about plans for studies, talking with vendors. So Kriya being a small company, a lot of the studies are outsourced to CROs or to other external partners who perform a lot of the animal work or the cell line work. And so kind of coordinating with those people to get these studies going and look at the data coming out of them. Now my days are going to kind of shift because we've done a lot of fundraising lately. We've raised a lot of capital. And so we're bringing a lot of those capabilities in-house. So we're starting a genomics group, which I'm going to lead. We're starting immunology. We're bringing assay development in-house. So we're hiring a bunch of in-house scientists. I know I'll talk about that later, but so now my days are going to be spent managing a lab, managing staff, still sitting in project teams, trying to drive projects forward.
That's very exciting. So for the graduate students and postdocs who are on the call with us today, what do you recommend that they do if they would like to work in this field too? What could they be doing now to explore and prepare?
Yeah, so it's great to talk with people who are in the industry that you want to join. I think also I did an internship as a postdoc. If I had known that was possible during graduate school, I would have probably done that during my PhD. I seem, before I did the internship, I kind of thought that, well, people in industry, as a data person, that I would just be in some closet basically, and they would feed me data, I'd analyze it, and I'd ship it back out. And I just thought that it was in this kind of, siloed position, and that it'd be like churning through data. But it's not like that at all. I'm very involved with the bench scientists, with other data scientists, with large project teams. And that's been the case at every company I've been at. So it's not a Kriya specific thing. But so I would say doing an internship to get a sense of what it's really like to work in industries, try to get an internship at a place that you could see yourself working in when you graduate, make those connections. That's probably the best advice I could give.
Terrific, thank you. So I think you've already talked a little bit about this, but the employment outlook at Kriya seems very positive. Do you have a sense of kind of the larger industry sector that you're in? Is the post-COVID hiring outlook pretty positive generally?
Yeah, so the biotech job market is really hot. People are having trouble hiring because there's just such demand for biologists right now, and for data scientists in particular. I think that I've been approached for data science jobs that have nothing to do with biology. And I told you, my first role at Novazymes was not even the type of data that I was trained to analyze. So it gives you a lot of opportunities in different markets. I don't see it slowing down. And I think it's great because, data jobs, they're flexible. A lot of companies now have embraced remote work because of COVID. So you can work from almost anywhere. And yeah, I think just keeping up data science skill sets. I will say, for those of you on the call that might not be data scientists, gene therapy, which is what Kriya, that's our platform technology. Gene therapy is also a very hot field, looking for people in expertise in molecular engineering, immunology. So lots of potential, I think.
That is excellent news. So let's move on to now our bonus 6th question in that light in terms of hiring and a positive outlook. Can you tell us about the PhD level positions that are coming open at Kriya?
Yeah, so I love that you asked this question because we have a lot, we're hiring a lot. So we just raised our Series B. Like I said, we're bringing a lot of our activities that were external, internal. So here in RTP is where our manufacturing and our development and early research teams are based. So we're hiring in the immunology group, we're hiring in early research, molecular biologists, people with protein experience. We're also hiring in analytical development. And then we have another site in California who is our technology innovation group. So that's where computational biology and bioinformatics sits. Though, like I said, data roles can be anywhere. So you don't have to be in San Francisco for that role. We also have our high throughput molecular screening group out in California. So if you are a molecular biologist with, engineering background or doing high throughput assays and you want to move to San Francisco, then that group would be perfect for you. And then of course, I'm starting a genomics group here. So we're looking for people with genomics and NGS data analysis skill sets. So lots of open positions. And I will say that, when I started at Kriya in April, there were, I think 60 employees. Now we're at 100. And they only have plans to expand. And so even if you're not ready right now to transition out of your PhD or your postdoc, 6 months from now, we're going to still be hiring. So it's great. It's a great company, and I encourage any of you of interest to reach out to me.
That is fantastic. And should folks reach out to you on LinkedIn? Is that the best way to reach you?
Yeah, you can reach me on LinkedIn or you can email me, whichever you prefer.
Okay, so I can share your email address with folks who are attending today to follow up. Happy to do that. I want to pause for just a moment and see if anybody has any follow-up questions for Jennifer. So it looks like we've got a question in the chat. I hear the term data science a lot, and I'm curious what that covers in a biotech company. And how is that different from computational biology?
Yeah, so companies use it in different ways. So at Novozymes, I was, data science, and, that meant doing statistics and programming. And at Sarepta, I was computational biologist, but really I was working with genomic data sets like RNA-seq data or ATAC-seq data. At Kriya, the difference is that our computational biology group is really involved with machine learning and kind of advanced computing, whereas genomics, like what I'm doing now, is analyzing next generation sequencing data. So every company, has a different way they like to phrase it. When you're doing your job search, I would really set up job alerts based on skills that you have. So, if I was setting it up, it would be like NGS would be something I would put as a term. But if you're more on like the machine learning side, then, you could put those terms in your search, but every company has a different way they classify it.
Terrific, thank you. I think that actually opens up to a couple of the questions that came in through registration too, and maybe we can try to get those answered.
Sure.
So for somebody who is applying for a data science job straight after a PhD, are there specific types of programming or statistical knowledge that you would recommend everybody has? Is there on-the-job training available? What's kind of the environment?
Yeah, so each company will have their own set of skills that they want you to learn. At Novozymes, they were big on JUMP, which is a SAS statistical package. So I learned, JUMP programming, but I wouldn't, and that was kind of on my own time at Novozymes. I wouldn't say that anyone should learn, JUMP statistical programming unless your job requires it. It's not really broadly applicable. I came in to industry with a background in R and like Linux scripting, and I think those are great tools to have. Kriya, the computational biology group is big on Python, and if you're more interested in like back-end development, pipeline development, Python would be what I would go with. But generally, places have been like R or Python. I haven't really seen places that have required you to know everything. Once you learn one language, you can adapt to learning something new.
That is very helpful. And I've got one more question for you, Jennifer, that came in through registration, and that is, were there any skills that you really set out to acquire during your PhD or your postdoc to get you ready for an industry career? I know that you mentioned you were really looking toward industry as a postdoc and you did your internship, but were there any skills that you were really aiming to grow in?
Yeah, not that I particularly set out to develop. I wanted to grow more in my genomics expertise, my computational biology expertise. I had a lot of that in graduate school, but I just wanted to learn how to apply it to more situations, which is why I did a postdoc. I would say one of the most important skills is communication. Why I think I've been hired at all of these different jobs is not necessarily because, oh, she knows R or she can analyze genomics data. It's that the ability to present your data to a wide audience, to, understand the pain points that the companies are facing and how you fit into that. So it's a lot about communication, I would say.
It sounds like you also have a strength in strategic vision and looking at what companies are going to need your expertise down the road and think about how to position yourself there.
Yeah, so I mean, some advice you could do now is if you're not quite ready yet to get a job, go ahead and set up your job filters on Indeed or LinkedIn for things that you think you may want to do, and just look at the jobs that are out there, the requirements they have, the skill sets they're looking for, and try to develop that. And then when you're ready to actually apply, whether it's a year or 2 years from now, you can say like, oh, I have all these things. So that's another thing you could do to prepare.
Fantastic. We've got another question that came in through the chat. Do you feel that your wet lab skills were necessary for your current position as lab lead at Kriya? Or do you mainly use your computational skills from later in your PhD and postdoc?
Yeah, so the wet bench skills, I haven't touched a pipette in, I don't know, maybe since my first year of graduate school or second year. I think where it comes in handy is that not many data people are able to understand experimental details that bench scientists are trying to work out. So if you've never, even though I haven't personally run every assay that the company's running, I have enough experience, even though it was only a year or two of my PhD, to understand the assays they're talking about, and how to set up those kind of studies and interpret results. So really it's about communication again, but I can only do that because I had that bench experience. But yeah, no one has offered me a job because of any bench skill. I don't list them on my CV. I don't, highlight them or anything. It's more about being able to communicate to the company that, look, I can analyze your data, but also I can work with the bench scientists to design their experiments and then interpret the data on the other side in ways that's relevant for them to understand. There's, with genomics data, with any kind of large data sets, there's hundreds of analyses you could do, right? But you choose to do certain ones because the bench scientists are trying to ask very specific questions. So like understanding what they want, understanding how your data fits into it. I think having a bench background is very valuable.
Thank you so much. All right, looks like we got another question that came in through the chat. This one from Lauren. I use Python on a daily basis, but mostly for data analysis using existing libraries, pandas, and notebooks. I can write some basic scripts and troubleshoot, but I consider myself a complete novice at Python. Is this an adequate level of experience for a job in data science? Especially, she says, I'm interested in working as part of a team to analyze results.
Yeah, so I guess it depends again on the job. Like I get the sense when I interview whether it's a position that I would enjoy or not. And I would not enjoy a position where it was heavy computation, right? Like I can write scripts, I can do things in R, but I'm not a computer programmer. And so if a company wanted me to take on that role, it would not be a good fit for me. And that's okay. I don't want to do that. At Kriya, what appealed to me when I first joined is that they do have a computational biology group. We just hired a bioinformatics infrastructure engineer who's going to manage the cloud environment. I can find my way around AWS, I can install things, I can run scripts, but managing it from like a production standpoint is not my background, and it's not what I want to spend all of my time on. So knowing that they were going to bring that expertise in-house so that I could focus on the types of data analysis that I enjoy, then that made the decision easy for me. But you will find, so you're going to find bioinformatics roles that are heavy on the computation, that are heavy on the back-end development. And you're going to find roles with the exact same title where they're not, as heavy. And you can glean some of that from the job ad. Like if they list, 20 packages that you need, extensive experience running or customization, you can kind of get a sense from the job ad, but also talking to people in that group or in that company to understand where your skill set really fits best, I think is the best way to approach it, or you would figure that out on an interview.
Thank you so much. Wow, I think that's very helpful. So for folks on the call, we have just a few minutes left with Dr. Israel. Are there any more questions you'd like to ask? And feel free also to use the raise hand feature if you'd like to come off mute and ask the question live. Rachel asks, where do you think the field of genomics will go in the next 10 years?
So it's going to be a lot of single cell sequencing, a lot of multi-omic approaches. So, in single cells, looking at, doing RNA-seq, doing ATAC-seq, doing ChIP-seq, doing all these different assays at the same time from the same sample in a single cell. That's going to become really popular. Ways to do high throughput proteomics is kind of the next frontier. Right now, we mostly are doing, sequencing RNA or DNA. Proteins kind of, not something we have really focused on. So that would be another frontier that people are trying to tackle. So yeah, those, and then more personalized genomics. There's all of these, platforms now for personalized care. And I think that's another big area.
Great, Rachel says thank you.
I've got a question.
Please go ahead.
So I know you mentioned that you were formerly in like Greg's lab. And I'm wondering if you can maybe talk or reflect a little bit on transitioning from like a relatively basic research interest to working in a company with primarily translational projects.
Yeah, so like I said, when I went to graduate school, it was, I wanted to do evolutionary genomics. I wanted to work in non-models, and that's still very interesting to me. But what's also interesting is that I think a lot of projects I do now have evolution angles to them. So there's actually a project I'm doing right now at Kriya where we have these different production cell lines for producing our therapy, and some perform better than others. There's a phylogenetic relationship known between the lines and the mutations that they carry. And the way I'm designing that study is exactly the same way I did my thesis research. So instead of, multiple sea urchin species, I have multiple cell lines. I'm doing time courses. I'm looking for, cluster shifts and expression. So, I think that the thing I love about genomics is that it doesn't matter if it's plants or bacteria or animals, doesn't matter if it's basic research or very translation based. The techniques are the same, and you can get really creative with the questions you ask. And I think, I don't know, I like being involved in a lot of different projects. Some are more translational than others. So like a translational project at Kriya, probably the most translational would be, sequencing vector genomes. So understanding if our therapies are mutated, or if there are certain, weird things going on in our vector genomes. That's a very downstream translation issue. But we also have very early research where they're just trying to understand, like, why is a certain cell type susceptible to treatment and others aren't, and so the companies have a range of projects kind of from that basic level down to very translational questions.
Thank you.
Yeah.
Thank you for that question, Riley. Well, I think that was a fantastic way to bring to a close our time together. Many thanks to Dr. Israel for making the time to join us today and talk about her career path and the many, many positions opening up at Kriya Therapeutics. I've just put a link to see those open positions in the chat for anybody who wants to follow that. And please join me in a round of applause for our speaker today. Thanks to everyone for joining us. We'll see you at the next Alum Zoom. Bye, everybody.